Now, what actually happens in the brain when alcohol is consumed? As we know, neurotransmitter systems do not function in isolation. That is why scientists are paying increasing attention to the integration of communication systems in the brain. Now let us look at the mechanisms by which alcohol consumption may affect multiple neurotransmitter systems to influence behavior. The connections between individual neurons are very complex. One neuron may connect with up to thousands of other neurons. Each one releases different types of neurotransmitters, and each receptor type responds to one type of neuron. Inhibitory neurotransmitters decrease the responsiveness of other neurons to further stimuli, whereas excitatory neurotransmitters have the opposite effect. Now, what are the effects of short-term alcohol consumption? If alcohol is consumed short-term, it depresses brain function by altering the balance between inhibitory and excitatory neurotransmission. Or more specifically, it increases inhibitory neurotransmission by decreasing excitatory neurotransmission. Now let us look at the main inhibitory neurotransmitter in the brain, gamma-aminobutyric acid, or short GABA. With a receptor subtype called GABAA, this neurotransmitter leads to a state of sedation and decreased anxiety. Some research suggests that short-term alcohol exposure increases the inhibitory effect of GABAA receptors. On the other hand, there is also work showing that it does not increase the effect of GABAA receptors in some brain regions and under certain conditions. Alcohol has also shown to increase the function of glycine receptors. Glycine is the major inhibitory neurotransmitter in the spinal cord and brainstem. The effects of alcohol in the part of this lower area of the central nervous system may cause some of alcohol's behavioral effects. Another possibility of how alcohol increases inhibitory neurotransmission is by increasing the activity of inhibitory neuromodulators, such as adenosine. This causes sedation, whereas inhibition of the adenosine system causes stimulation. Biochemical evidence indicates that in laboratory experiments, short-term exposure to alcohol increases the levels of adenosine that can interact with adenosine receptors. This could explain why alcohol acts as a sedative. Now let us look at another way how alcohol changes the balance of inhibition and exhibition. Alcohol might induce sedative effects by reducing excitatory neurotransmission. The two major excitatory neurotransmitters in the brain are the amino acids aspartate and glutamate, which act through both NMDA receptors and non-NMDA receptors. It appears that short-term intake of intoxicating concentrations of alcohol inhibit activity of those receptors, potentially resulting in sedation. However, these receptors are relatively insensitive to large alcohol concentrations under some experimental conditions. The need for further research is thus underscored. Another part of the body, or more specifically the brain, that alcohol impacts is the memory. Complex brain functions involved with the memory are controlled by a variety of neurotransmitter and neuromodulatory systems which all work together. Memory formation appears in the form of changes of how synapses communicate with each other produced by a sight or a sound. A fundamental part for memory formation is the phenomenon called long-term potentiation, LTP. LTP is a sudden but lasting increase in the overall level of excitatory neurotransmission in the hippocampus which is involved in memory. It seems that for LTP to take place, glutamate receptors must be activated and GABAA receptors inhibited. In some cases, short-term exposure to alcohol inhibits glutamate receptor function and stimulates GABAA receptor function in the hippocampus, thus inhibiting LTP. This theory has been backed up by other reports. If LTP does actually play a role in memory storage processes, alcohol's negative effect on memory could be related to its effects on glutamate and GABA systems. Previously we had only looked at what happens when alcohol is consumed short-term. But what about long-term alcohol consumption? While short-term exposure may increase GABAA receptor function, prolonged drinking has the opposite effect. This may result from a decrease in receptor levels or a change in the protein composition of the receptor, leading to decreased sensitivity to neurotransmission. This also works similarly with glutamate receptors after long-term alcohol exposure. After a certain amount of time, our brain builds up a tolerance for alcohol, which means that you have to drink more of it to attain the same effect. Under investigation, it has been found that tolerance is regulated by connections between neurons producing neurotransmitters or neuromodulators. Apparently, the pituitary hormone, which has effects on body fluid equilibrium, vasopressin, plays an important role in the maintenance of alcohol tolerance. A single exposure to a vasopressin-like substance while an animal is under the influence of alcohol is followed by a long-lasting tolerance. This tolerance does not only depend on vasopressin but also serotonin, norepinephrine, and dopamine. When alcohol consumption is abruptly reduced, something called alcohol withdrawal syndrome may take place. An increase in calcium that enters nerve cells occurs.